to before with the UPEC, and talking about its aerobactin siderophore, which allows it to sequester iron. Iron is super important in biological systems. It's essential for almost all living cells. It's obviously central to the function of many biological processes. We know that it's a cofactor in certain enzymes like superoxide dismutase or cytochromes in the electron transport chain and so forth. So it's really important, but it's very limiting. And one of the reasons, obviously, is the human body also needs those things, needs iron, and so it's very good at sequestering iron, so that it's not just floating around, so that microbes can grab it and grow like crazy in your blood. Most of our iron is bound up in hemoglobin, but some of it's free in the form of Fe3 plus or ferric iron. We have to combat microbes in their ability to acquire the iron, and so microbes that produce these siderophores will be often better off bloodborne pathogens if they're able to grab the iron. There's also another strategy that microbes use called iron abstinence, where microbes like Borrelia, the guys that cause Lyme disease, for example, have solved the iron problem by using manganese, for example, as an alternative to iron. And so they'll have a lot of manganese cofactored enzymes as opposed to iron cofactored enzymes. Not surprising, because we know that iron is so important. There's lots of really interesting iron uptake systems that bacteria have evolved that are dependent on contact between a bacterial receptor and some kind of host iron binding protein, or an iron binding molecule that's not a protein. Typically involves specific receptor protein interactions, and so again, they're often energy dependent, and there's usually a few factors involved, like a permease and a periplasmic binding protein and so forth. But what's cool is that a lot of these iron systems dictate the host range for particular organisms. So Neisseria, for example, are primarily good at grabbing human iron, and so they're most adapted to living with humans. Haemophilus influenzae, the microbe that can cause ear infections, but also meningitis and so forth. But this is an organism that has a wider range of whose iron it can steal. You see human, bovine, and rabbit. And Bordetella pertussis, the microbe that causes whooping cough, has an extremely broad range, and so can infect a lot more different organisms. Transferrin is an iron binding protein that you and I would produce to sequester our iron. And so in this particular case, you see receptor-mediated iron uptake in a bacterium. In this particular case, a gram-negative bacterium. We can tell because it's got an outer membrane, the periplasm. So the cell wall would be in here, and then the cytoplasmic membrane. So in this particular case, there's a host iron binding protein. So transferrin, for example, is one. And here's the little iron right here. And so this microbe would have an iron binding protein receptor here and would bind to the transferrin iron complex and then pluck that iron off and then pass that through a lipoprotein in the periplasm, a permease in the cytoplasmic membrane that's complex to an ATPase, hydrolyzing ATP and then spitting the iron into the intracellular iron pool. This is a slightly different version, but same end result. But in this particular case, it's siderophore mediated, so like the aerobactin system that I already mentioned. So the microbe produces these little hexagons here that go out into the world. These are the siderophores. The siderophores would then go out and knock the iron off of transferrin and then come back almost like a boomerang to a siderophore receptor. The iron and siderophore are then brought into the intracellular pool. The siderophore is obviously recycled and sent back out into the world to grab more iron. And then obviously the iron itself is used to, again, make iron cofactored proteins. TonB is a stabilizer, but may help with proton motive force. I don't think we're all that certain what TonB does, but it's definitely part of the structural apparatus in these siderophore-mediated iron uptake systems. So here's a great way to look at siderophore activity on plates. So this is Cass agar, and it's for siderophore detection and culture. And so you have two different organisms. One's a wild type strain that produces a siderophore, and one's a mutant that lacks the siderophore. The medium itself has iron complex to a dye. And so if the bacterium can make a siderophore that goes out into the world and grabs iron and comes back, it'll pull the iron off the dye, and the dye turns orange. Normally it's blue. And so you can't really tell here, but normally it's blue. And so when the iron gets pulled off, the dye turns orange. And so you see a nice halo around here showing how this organism here was able to produce siderophores into the medium and grab iron and change the medium.